introduction, and very much thanks for the opportunity to share our work in the past few years. So I'd like to start with this figure that from the editorial from the European Heart Journal that comment on our recent publication in the same issues, because I find this figure is quite informative to illustrate the idea that I want to present today. So in epidemiology, we want to have two goals. One is to understand the mechanism underlying disease. We want to find out the causal risk factors. And we also want to do prediction in terms of disease risk or treatment efficacy. So these are the two big goals for us. In traditional epidemiology, we look at, say, questionnaire-based information collect from the individual, for example, assessing their dietary intake, and then we link those risk factors to the disease outcome or phenotype of interest. Right now, with the affordable biotechnology, more and more omic dataset like metabolomic, microbiome, genetics, and transcriptome and others become more and more affordable in larger scale epidemiology studies. So right now, many of the cohort has maybe 2 or 3 or even more omic data sets. With this, we now can have an objective measurement of this epidemiology data set, for example, including individuals' exposure to environment, to lifestyle, dietary habits, and then we can link those to the phenotype of interest. And more importantly, we can utilize this omic data set to understand the function and also the biological response for individuals to those impact from environment or genetic impacts. So the question now becomes, how are we going to integrate these different omic data set to serve the purpose to identify causal risk factors and also to help us to do prediction. So I hope you have done your lunch. If not, this hope can give you some flavor. So this question is like, if you have all the food ingredient available, can we have a program that just intake all this omic data set and then give us interesting findings? It's like if you have all the ingredients from this seafoods, they just put in a magical pot and then just cook it, and then you can give you a very delicious food. Right? I think the question to this would be probably not such a magical pot would exist. So my answer to the previous question would be, there isn't such of the program that you can just magically put all these omic data set together and then we output in interesting findings. So integrating omic is very hypothesis driven. We need to really work with the scientists to understand what is the goal of the science problem behind. And I summarize that there are two dimensions in the integrative approach. One is that the omic will bring us the new territory for novel biology, because its comprehensive measurement, say genetic on the whole genome, metabolize for a large category of small molecules, proteomic with many of the proteins. So we are able to identify new biology with this comprehensive assessment of a specific function. And with that, many of those might be relevant to the disease outcome of interest. They might have small effect size, but combining those effect size turn out to be quite useful in terms of predictions. Even though they are not surviving this multiple hypothesis testing, so these are useful for prediction. So they serve the goal for both in terms of identify causal risk factors and also the prediction. Specifically, the results that can bring from multi-omic, including you can use those to understand the functional cause and the downstream effect of the exposure of interest. We can use those to link disease and trace with common genetic effect. They could be either a pleiotropy scenario or they could be linked by a causal link. But the association we find is quite robust in terms of environmental confoundings. And then they can help us to partition phenotype heterogeneity into specific pathway. I'm going to talk more about this later on. And then they also provide an objective measurement of exposure, less subject to record bias and other measurement errors. So when I think about how we model or to formulate the statistical analysis when we integrating multi-omic. This is the diagram that I would like to use, and this is the follow from the mediation frameworks. I would think the environment and the genetics are on the outside because the genetic is not changed. We are talking about germline genetic mutation here. And then other omics, they are like serving as intermediate phenotype. They can mutually affect each other. And eventually, they affect the risk or the traits that we are interested in terms of the phenotype. And we have many, many phenotype available. So this is another kind of omic that called a phenomic that we could be interested. So this shows the breadth and the diversity of the omic data set that we will be able to work on. But another dimension that's often neglected by the research community is the time scope of the impact by this different kind of omic. For example, if you talk about the life course of an individual, and genetics actually cover the whole life because it does not change. We are talking about germline genetic here. If we are talking about risk, this is about the lifetime burden of the risk. On the contrary, epigenetic like DNA methylation, it could be quite stable over a few years. How stable it is actually depend on the tissues, depend on the genes and the trigger. As I learned from my colleague, some of the heavy mental exposure could rapidly change the methylation on certain genes. So this depends, but it's kind of medium-term impact. And metabolomic is more of the short term, right? Within 48 hours or a few days, the metabolism would complete for a certain metabolite that we intake or the food that we intake in our body. So it's more of the short term impact representing a short term. So I think we need to take into account the dimension of time when we talking about integrating multi-omic. And especially when we talk about the timing for the screening and the intervention, and the timing for the disease onset and to evaluate the efficacy of the treatments. So I'm going to talk about these three omics one at a time, but you can see that they are not mutually exclude from each other, but in fact, they are supporting each other so that we need to integrate them. So first, I will talk about our research on genetics. So linking multi-omic biogenetics, because the natural direct effect from the genetic mutation, germline mutation, to the disease phenotype of interest, that is a natural direction. It's not subject to reverse causation, and the confounding is arguably only due to population stratification, but we can account for population stratification pretty good using genetic data itself. So the association between the genetics and the disease is quite robust. And if we can link two diseases using the genetic link, then the association between the disease and trace would become robust and not subject to environmental confoundings. Right. And because in the last few years, the larger scale biobank data provide a very powerful results for genetic association study. So now it provides more precise estimate of the genetic effect that we can borrow from, which is not feasible in maybe several 5 years ago. And summary statistic-based method allow us to overcome the hurdle for data sharing and privacy protections, because we only need to share the summary statistic in order to benefit from the larger scale GWAS without sharing individual level data. So these are the advantages that we can utilize by linking multi-omic using genetics. So just to give you a sense about the power that we can gain by recent GWAS. For example, this is a paper we look on childhood asthma in 2007, published on Nature's. We only have 300,000 SNPs, about 1,000 cases and controls. This is the chromosome 17 regions or OMDL3 regions that we identify at that time. But in 2018, even based on the early release from the UK Biobank, we have about 8 billion SNPs to work with, more than 10 times the cases and 70 times the controls. And you can see that many of the other locus can be identified with the asthma. And the chromosome 17 region is still here. The GSD-MB regions and ORMDL3 region is right here. So, but you have more power to detect other important regions. Okay. So first, I want to illustrate an example to how to integrate disease and expression, so transcriptome, using genetics. This helps us to interpret the function of the genetic variant we identify from the disease. For example, in this paper in 2007, we identified a childhood asthma locus at chromosome 17 with childhood asthma. But in this region, we have 35 genes. We don't know what is the functional gene for asthma. But integrating the genetic effect on the gene expression, we were able to identify the ORMDL3 expression is highly correlated with the same genetic variant for asthma. And that led to the identification of the ORMDL3 expression, ORMDL3 gene as the candidate gene. And this has been shown in the follow-ups many studies that to show the causal role of ORMDL3 with asthma. This is feasible because the genetic variant on gene expression is a high power. We only need a few hundred of the samples to detect these associations. And therefore, even though from the genetic variant to disease, there's a big step unknown for the function, we now can move one step forward to understand the function of the genetic variant that identify for disease. So by integrating gene expression and genetic data, we were able to interpret the function of the genome-wide association study. And because this strategy is so useful, so we later on developed the genetic for expression and also genetic for methylation, for DNA methylation in later studies. And we curate this database so people can utilize in their own study and for different disease. So as you can see, even though this EQTL are based on whole blood data set, but when we apply to other disease, it's not just for asthma. It could be useful for many other immune disease or inflammation trace and others. You can see I only list those that I'm directly involved contributing the EQTL analysis in those. You can see it's cover a full range of disease. Right. So this is disease and expression integration. With the past few years, when the larger scale biobank become available, we start to think about, can we integrate disease and disease genetics by you doing this cross-trait genetic analysis. There's a two big advantage. One is that we can utilize the genetic link to avoid environmental confoundings. As I said in spring before, you can utilize this linking from the same genetic variant on the both disease. If they do have the link, then you can say these two diseases have a robust association. They might share the same pathway, or they might be in the causal direction from one to the other. And we also find that this is useful to link different diseases to understand heterogeneity. That I'm going to show you as example using asthma. For example, asthma is a highly heterogeneous disease with the allergy spectrum of asthma and also non-allergy asthma. So if we try to understand what is the pathway or genes that are responsible for asthma, allergy asthma, and what is the gene or pathway responsible for non-allergy asthma. Then we can do cross disease using asthma as the primary trait of interest, and then cross with allergy disease, like including hay fever, atopic dermatitis, and other allergy symptoms. Then we can see what is the overlapping genes for allergy spectrum of asthma. And if we do a cross-trait analysis with obesity or cardiometabolic traits, then we can try to understand what is the non-allergy spectrum of the asthma. We also find interesting cross-trait genetic between mental health disorder, including ADHD, major depression with asthma, and that we will be able to use those overlapping genes to understand the different heterogeneity that are leading to different kind of asthma. So this seems to be a useful strategy that we can utilize. So taking the asthma and allergy as example, we can see that if you're looking at genetic correlation or at the whole genome level, which is the number shown on the upper triangle, you can see the asthma has a substantial genetic correlation with allergy disease, but not that much for rheumatoid arthritis, Crohn's disease, and others. So this is at a global gene level, we can understand how share genetic between different disease and traits. And after that, you can focus on the disease and trait that do share genetics and then narrow down to individual variants. Say these are the genes that are shared genetics as specific genes between asthma and allergy disease. With this, if you further integrate this with the gene expression data from the NIH Genotype Tissue Expression Project, that we can see what are from among these shared genes between allergy and asthma, what are the tissues that are overly expressed for those shared genes. So we can see what's the functioning tissue for those genes. And not surprising to see skin and the lung are among the top the genes, and other tissues that has the epithelial cells are also among the enriched tissues. So that tells us what really are the functioning tissues and what might be the candidate tissue that we can follow up in the functional experiment. Okay. So once you establish the link between the disease, the next question you might ask, are they pleiotropy, say the same causal variant affecting the two diseases, or they are related to causal to each other? For example, anxiety and asthma, we found they have a substantial genetic correlation, but does not seem to be causal to each other. So this is the result suggest from the Mendelian randomization analysis that asthma and anxiety don't seem to be causally which one has an upstream effect or to the other one. And but on the contrary, if you're looking at BMI and asthma, we find strong Mendelian randomization results to suggest BMI is the upstream effect on to asthma. Instead of asthma causing BMI, is the obesity more likely to cause asthma. Right. And then we find this strategy to be quite useful, not just for asthma and disease, but also for many other diseases. We look at Alzheimer and cancer, Alzheimer and glucose and insulin metabolism, and also testing diabetes and resting heart rate is another interesting finding that we find genes at the nerve tissues and immunology tissues sharing between the diabetes and resting heart rate. And recently, we look at COVID-19, severe COVID, hospitalized COVID, and the non-allergy asthma seem to increase the risk for severe COVID-19, and obesity and its genetic score is also has an increased risk for the COVID-19. And recently, we used the China Kadoorie Biobank to look at lung function and obesity. So we find this strategy to be quite useful for many different kinds of disease. We also find that not just for identify causal variant, but also utilizing the cross-trait genetic, we were able to boost the power to predict the primary trait of interest. Say here, for example, trait 1 is the trait that I'm interested to predict. If we find the different trait like trait 2 and 3, they share different genetic with trait 1, we were able to incorporate this information in a machine learning model that we will be able to boost the power to do a better prediction for trait 1. Right? And the algorithm can automatically select which trait could be useful and share genetic with the trait 1 of interest. Right? Because this is very large scale computation, we design a multi-CPU, multi-core distributed algorithm so that we can analyze this data on the high performance cluster on the hardware and also implement on the Google Cloud. Right? So it's become doable. And just to show you some results that we utilize this model to predict height. So human height is our primary trait of interest. By incorporating genetic for BMI, we were able to boost the power to predict the height. For example, the gap here shows the multi-trait model compared to the single trait model. You can really boost the prediction accuracy from 37% to 42%. Just to give you another idea about prediction accuracy, if the prediction should be useful in terms of clinical application, I think a well-calibrated method would be quite critical. LDPRET is one of the methods that has been widely used in building polygenic risk model, and this is the single trait model. And as you can see, predicting the actual high using the genetic score, the R-square is already 35%. But if you look at the men and the women's individually, the two groups, the slope is not close to 1. And because the machinery would optimize the performance on overall prediction accuracy, but not within group. So the individual prediction value is not very well calibrated. But if you can boost the power using the cross-trait model, the CTPR, you can boost the R-square to 42%. Although the 7% gap does not seem to be extremely large, but if you look at individual predicted value, it's more well calibrated. So I think going forward, if we can build an even better prediction model, then it could become more and more useful in terms of in the clinical settings. So we summarize this approach in a review paper that it consists of multiple steps because we think this approach can be useful for many different kinds of disease. It consists of 3 steps. You need to curate the GWAS for different disease of trait of interest. And then you can look at overall sharing between the traits and then identify which trait would be your target to work with. Then you can do cross-trait analysis to identify the variant that is responsible for the sharing of the genetic and the functional consequence, and also integrating transcriptome to understand what are the gene or tissue that might be relevant for the sharing, and then using Mendelian randomization method to infer the potential causal relationship or the prytropy between the trait or disease of interest. So I think this could be extended to many different kinds of disease. And now I'm going to switch gear to epigenetics. So this is motivated from around 2014-15 that we work on genetic for total serum IgE, which is just a trait for allergy. And at that time, the genome-wide association study do not identify much useful drug target for IgE, and the top GWAS variant only explain about 1 to 2% of the total variation in IgE. So we start to look at whether epigenetic like DNA methylation can be helpful to identify interesting gene for IgE regulations. So we do a so-called EWAS, the Epigenome-wide Association study, using multiple cohorts from the UK and also the Canada. And this shows the top hits around the genome. You can see the quite significant associations, and the QQ plot indicating strong association and not much of the inflation due to potential confounding. Because now we are looking at epigenetics, it is not. It is subject to reverse causation, unlike the genetics, right? The genetics is not subject to reverse causation, but here it could be. So we should be more careful about potential confoundings here. And one challenge of the epigenome-wide association study is that, say, DNA methylation has different impact or different level across different cell type, and we can only assess to whole blood from the larger scale epidemiology study, and they have consist of multiple cell type, and different cell type have different level of methylation. So cell type could be a confounding for this association. So before we conclude is the methylation that associated with the outcome, we need to rule out the possibility for confoundings. So here, eosinophil is one of the cell types that's relevant to IgE. You can see if we color the individual based on their eosinophil counts, they have a shifting from the y-axis here, right, up and down, and also the shifting from the left to the right. So indicating methylation have different level between eosinophil and other cell type, and IgE also is associated with eosinophil counts. Right? But doing this stratifying analysis, it suggests this association does not go away, does not explain away by eosinophil counts. And using Mendelian randomization, we integrate the genetics for methylation. So we have the methylation QTL data available for between the SNP, the genetic variant, and the methylation at IL4, for example. we do see that causal effect from IL-4 methylation to IgE seems to be quite robust and statistically significant. And using a mediation framework, we can decompose the association between IL-4 and IgE and see how much of these can be explained away by the eosinophil counts and how much cannot. We do see the direct association to be quite robust. So all these statistical evidence suggests it is the eosinophil, it is the methylation in the eosinophil that's related to IgE. But in order to confirm this, we actually need to do this functional experiment to collect eosinophils extract from 24 individuals, including a asthma patient with high IgE, a asthma patient with low IgE, and a healthy control. We do replicate it. The methylation and IgE do have a negative correlation, and this is the methylation within the eosinophil cells. So tissue-specific methylation is confirmed based on functional data set. And if you look at the methylation level within eosinophil and others, you do see this methylation level has a lower level compared to other cells, like including lymphocyte, whole blood, neutrophil, and others. So it looks like these genes are turned on because the methylation level is small, it's lower, and in eosinophil. So this is a typical situation because we now have both potential confounding and potential causal effect that's related to methylation within eosinophil. So this is a challenge. But we can use this integrating approach to identify this causal association and validate by this functional experiment. So at the end, we find this top CpG related to IgE, and only the top 3 can explain 13% of the variation in IgE. And the top genes are potential drug targets. So to be more exciting, we now are able to follow up these findings in a longitudinal prospective cohort that we are obtaining the funding to do DNA methylation collect from 2 cohorts, one with the kids with bronchiolitis at the first year of life, and the other is the healthy kids. We are going to measure their methylation within the first years and also at 3 years. We will be able to look at how methylation is related to IgE sensitization at age 3 and obesity at age 3 and eventually asthma at age 6. So just to do some prior examples showing the IL-5R top gene from our previous study, based on 94 individuals already show interesting interaction effect with the IgEs and DNA methylation from the infancy on the risk for 5-year asthma. So we are quite exciting to follow up on these projects. And we also have the method available to account for potential confounding that we do not observe in an epigenome-wide association study. By fine-tuning these methods with existing method, we show that the smart SVA approach that we developed can well preserve the false positive and also can have a good power to detect the causal association. We also have the method to deconvolute the methylation within specific cell type, because if you only have the methylation from the unsorted tissue, say the whole blood, and if you know the cell proportion, you can, using a deconvolution model to estimate the methylation level within individual cell type, and then test for the association between the cell type specific methylation with the outcome of interest. So the estimate value showing up on the left and the observed value based on experiments seem to be quite consistent to each other. So you can recover the variation, for example, IL-4 methylation within eosinophil using the same data set that we used before. So using DNA methylation and epigenome-wide association study, we will be able to identify individual variants for the disease. I also want to highlight that using methylation, we will also be able to use this as a composite biomarker to assess the effect on aging. For example, using a methylation clock, we can assess how environmental impact or our body composition can affect this agent biomarker. We tried, when we started to look at this, we find out the existing marker or existing methylation clock developed by a European sample does not directly applicable to the Chinese sample. So we developed a Chinese sample specific clock, and using the data from our collaborator in China, it shows the correlation between the true age and the methylation age is 0.95 using these healthy individuals. It looks pretty good. And then we try to replicate this in another healthy cohort in the same province. And it also showed the R is 0.94, so it's pretty accurate. And then another replication using data from Beijing is actually coming from 4 different provinces with the sample from obesity and healthy individual. The correlation is 0.96. So again, it's replicate pretty good. And then we apply this to a Canada sample with the European ancestry family data with asthma and healthy individual. The correlation is, again, pretty good. So we are pretty confident that this can be applied to not just for Chinese sample, but also for other populations. And recently, we applied this to an intervention trial in the Israel. And we also see the correlation is 0.93. So looks pretty amazing, right? When you apply to across the world, and looks pretty good. And if we apply this to a data set with the cold oven workers, where they have been highly exposed to air pollution and chemicals, we see a shift of the methylation age upward more than a year, shifting upward. So it looks like their clock going faster in terms of their methylation. And if we look at individual PAH, which is the chemical that we measure from the urine, we can see that methylation age do positively associate with the high exposure of this air pollution. And if we apply this model to the Israel intervention trial, we can also see that for people have successful weight loss or have a healthy liver, percent fat, they seem to have a slower methylation clock after the 18 months intervention. So the clock seems to going slower if they do achieve some successful weight loss and maintain a healthy liver fat percentage, comparing to those with fatty liver and those with weight gain or unsuccessful weight loss after the interventions. So those are what we have found based on methylation. Now I'm going to switch gear again to look at metabolomic. So our start is to start look at this when this LC-mass-based metabolomic become available in large scale cohort. Here the first project, we try to look at how metabolomic profile can help us to measure the intake for dietary intervention and also the response from individuals. So we start from this using a Mediterranean diet based on the Predimed intervention trial in the Spain. This is a measure by 14-item score, so measuring how much they eat and how the higher score were indicating a close follow of the Mediterranean diet. And we try to build a model using the 300 metabolite to represent this. The idea is coming from the observation that the reported score consists of the true intake and also the reporting error. If we regress the reporting intake on the metabolite, we will be able to regress out the reporting error because only the true intake can affect the metabolite. The error, the record bias do not affect the metabolite. And if that's the case, then we will be able to represent the true intake better than the metabolite. Right. So that's the motivation. So we use the Predimed baseline to build the trainer model and then replicate in the Predimed year 1 metabolite and also the nurses health study and health professional study, which also have the metabolite and generate from the same lab at the Broad Institute. So looking at this model consists of 67 metabolites for this Mediterranean diet index. We can see that it's replicate quite well across the year 1 of the base Predimed sample and also in the nurses' health study, the correlation between the reported score and the metabolite signature is 0.3. So it's pretty robust across Europe, across the Spain and the United States samples. And the metabolite that select into this model seem to be quite relevant to the food intake. They're high in terms of fish oils and others amino acids that relate to Mediterranean diet. So that's pretty promising. So then we go on to see, is this instant score really predictable to disease? So by comparing to the reported score, you can see that the reported score and the CVD outcome do have a protective effect, right? But after adjusting the metabolite signature, the effect size being attenuated. But if you look at the metabolite score, it's still associated with CVD outcome with a stronger effect size. Even adjust for the self-report score, it does not attenuate much, right? So it will remain robust. So that suggests the metabolite score seems to be related to the disease outcome better than the self-report score. And the same thing can be replicated using the year one metabolite and the incidence CVD in the same cohort, and also in the nurses' health study and health professional study. So this also replicate in the states samples. So now we're trying to understand what are the variation, say for people that report the same intake, same level intake, they do have a substantial variation in terms of their metabolite score. Why? Or what are the causes that driving this this variation? We find that different food composition might explain 4 to 15% of such a variation. Because people reporting a score of 9, they might eating different food, but they still end up with a score of 9, right? And we also find genetic could explain about 12% of the variation in these variations. So this gives us an opportunity that we can now integrate genetics into the metabolomic studies. By doing a GWAS on the signature score, we find some top genes. For example, FADS1 to FADS3 are the fatty acid metabolism genes as one of the top genes that associate with the signature. And it's highly expressed in the different tissue, relevant tissue, like the brain, artery, heart, and adipose tissues as well. So this is leading us the opportunity that now we can use Mendelian randomization to assess what's the potential causal effect from the metabolite signature to the CVD outcome. And we do see the significant effect, the potential causal effect from the signature to CVD outcome by integrating genetics with the metabolomic here. Okay. We can do even further by looking at risk factor for CVDs. And using a multi-step Mendelian randomization, we can link the metabolite signature to the risk factor, including BMI, lipid, and diabetes, all the way to CHD and stroke. You can see in all the steps, they are significant. And the effect size comparing to the reported score, the effect size for the signature is usually larger than the self-reported score based on food frequency questionnaire or based on the self-report, the 14-item score. So this seems to be suggest this signature could be a useful way to assess the intake as well as the response, biological response for the Mediterranean diet. It is also useful in terms of predicting the future risks for CVD. Okay. So when we are working on this multi-omic study, we always hear about some of the questions about, for example, why do we need precision medicine? Why do we need precision intervention? For example, why not just give statin to everybody so that we can prevent heart disease? Statin is not expensive, right? So it lower your lipid. And why not ask all the food company to reduce sodium, right? Why you have to look at the individual and then ask for individual advice for sodium intake. Why not just ask the company to reduce sodium? And why we have to spend hundreds of dollars to get the genetic assessment, but why not just ask everybody to exercise more? Because exercise is very healthy. Eating healthier is also healthy. Why you have to do a polygenic risk model? So we try to approach this kind of question. I think this is all very important questions. We find that we can approach this by integrating genetics and omic data together. For example, in this study, we published on eLife early this year. We try to look at if we compare the effect of statin and 5 healthy lifestyle, we can look at how their potential impact on their lipid profile based on NMR. Because it is unethical to ask everybody to take statin, so here we used a genetic approach to represent the effect for statin. We used the China Kadoorie Biobank, which has 500,000 individuals collected, and also they have 100,000 with GWAS and follow-up, and their effect, their disease risk are also followed based on insurance records. So they can trigger if someone report disease, they can be report back to the this biobank. And previous study in the same data also show the 5 healthy lifestyle can well protect major cardiovascular or coronary events. So here we are looking at how these 5 healthy lifestyle can affect their lipid profile based on 4,600 samples with the NMR lipid profile. And this is based on a nested case control study with CHD stroke with the also common controls. So to mimic the effect from statin, we use the genetic score because the target for statin is the HMGCR genes. So there's a previous publication on New England Journal of Medicine in 2019 also showed that genetic score on upstream level gene for ACLY can represent the effect for potential drug target similar to statin. So we compare this approach and apply this to the China Biobank. But as you can see that the genetic score based on European SNPs and European weights does not seem to work well in the Chinese sample. But if you using the same gene, but selecting the different SNPs on the same gene and building the same way, different way based on the Chinese sample, you can substantially boost the power to represent these genes. Right? And this is evaluating based on an independent sample with clinical measure LDLs. So we utilize this as an instrument to represent the effect for statin. Right? So here, when we compare this to the impact on lipid profile, you can see the NMR provide us the size, the cholesterol concentration, and the triglyceride concentration in different lipids. And we can see that comparing to statin, so the red is the one with the taking statin, and the green is the one that taking both statin and healthy lifestyle, and the blue are the one that has a follow healthy lifestyle but did not taking statin. You can see the effect for healthy lifestyle is mainly on VLDL and HDL and also triglycerides content concentrations. Right? But for statin effect, it's mostly from for LDL, but not much effect on VLDL. So it looks like the lipid lowering drug and healthy lifestyle, they have a complementary effect on healthy lipid profile, affecting differently on VLDL and HDL and LDL. So it looks like you probably can benefit from both. Okay, so this is the finding that we found. So going forward, I think integrating multi-omic, considering their timing, is important. And it provides an opportunity for timing for intervention and the frequency for intervention as well. So for example, looking at obesity score, say polygenic risk for obesity based on their genetic data, you can see that individuals have a high genetic risk for obesity. They have a high fraction to go to become obese or overweight before 65 years old, but still someone would become normal at 55 years old. For individual with lower genetic risk, they are more likely to become normal and seldom become obese, right? But if you look at individual still are already normal at less than 65 years old, most of them would remain to be normal after 65 years old. Only about 20 to 30% would become overweight. So it's very important that you act earlier to maintain a healthy body weight before 65 years old in order to maintain a healthy body weight after 65 years old. And this is confirmed by our genotype and healthy lifestyle interaction analysis. So we look using the alternative healthy eating index as a way to assess the effect for healthy dietary intake. And if you're looking at cumulative healthy dietary intake, you can find the interaction effect is stronger if the individual adhere to the healthy lifestyle longer. So, and if you're looking at change, so how much change in BMI in terms of as an effect of change in dietary change to a healthy lifestyle, you can see the interaction effect is stronger if the change occur earlier in life. So that suggests if you add earlier to change your healthy lifestyle and stick to it for a long time, then you can benefit. And the one with higher genetic risk for obesity would benefit more compared to other people. So acting earlier is important. So who can be earlier? The baby, right, is the earliest if you're talking about action based on the genetic risk. So here with another project we established using a birth cohort, we will be able to integrating the multi-omic using in this birth cohort. We can look at the multi-omic in the maternal exposure and also the multi-omic in the core blood for the baby. So we can assess how this multi-omic can assess the longitudinal health outcome for the baby from birth to follow up to 21 years old. So this is a quite exciting project. And some preliminary results already show that some metabolomic network in the core is associated with the longitudinal obesity trajectory. For example, some individual become high BMI at very young age, like 2 to 4 years old, and maintain to be obese up to 16 years old. Right? So different individuals have different trajectory and they seem to be related to the core metabolite. So this is quite exciting ongoing projects. So going forward, I'd like to summarize that it's important to integrate multi-omic with longitudinal cohort. And for the young kids and as well as for adults with multi cohort, we can utilize. We have the data available, and also integrating large scale genome-wide cross-trait analysis can help us to bring into summary statistics from other cohorts as well. And we are extending this to our omic from proteins and also the virus in the neurology projects. So that's quite exciting. So hopefully we will be able to identify interesting findings in terms of prevention or treatments for this disease by integrating multi-omic. So finally, I want to acknowledge first the findings that to my projects for the 0R1 for the longitudinal cohorts and the pending grant that we get a promising score that we will establish food biomarkers using a feeding child. And also the findings for many of the collaboration projects that enable my group to work with many different kind of disease. and also the fundings to my group members, for my students and postdocs and other advisors that support their career development and recognize their contribution to the field. And it's grateful for me to be able to work with many colleagues here at the school in the Longwood medical area, the States, and as well as international collaborators, especially the program of the genetic epidemiology and statistical genetics. It's really like a family that with all these people support each other. So I really learned a lot, and I'm grateful to all the advice that I received and the collaboration that are ongoing and past collaborations. So really appreciate it. And finally, all this work is done by group members in my group, the postdocs, students, especially also the master student from the biostat and the epidemiologists. They're really tremendous people to work with, and they finally have a very good outcome also in their career. So I cannot list all the names here and their pictures. So yeah, so this is I'm very grateful to be able to part of them. So thank you very much.